This is in continuation with the earlier video on antigens. In that video, we have seen that antigens are substances which has an effect on the lymphocytes. They can stimulate the lymphocytes and produce antibodies or activated cells. Now, we are going to look at some of the works that were carried out in 1939 by Morgan et al. In trying to understand the types of antigen. We will focus therefore on the types of antigen that are available in nature. If you look at antigens, you will rarely find antigen in pure form. Antigens are normally associated with the bacteria, they are associated with the viruses, protozoans, fungi. Maybe there are antigens which are self-antigens. Our own cells will be producing antigens and we will respond to these foreign substances. Sometimes antigens are, you also find them in combinations with nucleic acids like nucleoproteins. You will find them associated with the carbohydrates. So therefore you will see varieties of combinations of antigens. So they are not found in isolated or in pure form. Antigens are known by its origin. You know that antigens can get into our system. It can be through ingestion, or it can be through oral system, it can digestive system, it can get inside, or it can be inhalated. So like several substances like chemicals or pollens, all these are other types of antigens along with the antigens of the microorganisms that enter into our body. Let us look at the general antigens that are present on a bacteria, for example. So for clarity's sake, it is drawn on the board. The kind of antigens that you find in a bacteria, first of all, let us look at, this is an H antigen. The H antigen is the flagellar antigen. Flagella, you know that flagella is composed of a protein called flagellin. And this flagellin is a very, very strong antigen that can cause a lot of, that can induce immune response in our body. For example, you look at the enterobacteria. So the enterobacteria has got the flagellar antigen, and which is very, very strong. Remember, when we carry out the Vidal test, we look for the H antigen and the O antigen. So that is the flagellar antigen. Another kind of antigen is and remember, the flagellar antigen is easily destroyed by heat, dilute acid, so it can be easily destroyed. Another kind of antigen is the C antigen. The C antigen is the capsular antigen. So in the capsule, all those bacteria with the capsule, it is a kind of a protective layer. So the capsule acts as an antigen in many cases. Another antigen is the extracellular antigen. So the extracellular antigen is the antigen which is the metabolic end product of a microorganism. So it is released into the surrounding that can act as an antigen. Another kind of antigen which is not shown here is the fimbrial antigen. So in the fimbrial antigen, which is also similar to the H antigen, it also can be very potent, and certain microorganism will release the fimbrial antigen. Another antigen is the K antigen. The K antigen is also called the capsular antigen. So it is the internal antigen. The K antigen is around the cell. Now there are different forms of the K antigen. Roughly about 50 different types of K antigens have been identified, which is highly resistant to heat denaturation, highly resistant to that. Another antigen is what we call the O antigen. The O antigen is especially the O antigen is the lipopolysaccharide. You know that lipopolysaccharide is used especially as an adjuvant in inducing immune response. So this O antigen is also a very potent antigen. There are roughly about 75 different types of O antigen has been identified. Another type of antigen is the I antigen. I antigen is the internal proteins of a microorganism. So when this microorganism is destroyed, the internal content can be released, and that can act as an internal antigen, which is also very, very potent. So these are the different types of antigen that you find, especially as an example, we have taken a bacteria as an example. So all these antigens have the capacity to induce an immune response in us. Apart from this, you are aware that there are antigens like blood group substances, which is also an antigen present on our body. So in all our blood cells, all our RBCs, these blood group substances are present. They are glycoproteins. So these glycoproteins becomes a very good antigen. So you are familiar with the difficulties that we encounter in blood transfusion, etc., the incompatibility because of the presence of blood group antigen. So this gives you an idea about the type of antigen that is purely determined by either species or based on the origin. There are also heterogeneous antigens. So heterogeneous antigens are those antigens which are found in different species. For example, in pneumococci or in chicken, there is a kind of antigen which is very, very similar to the blood group substances. So these are the heterogeneous antigens which are found in different species. We use antigens mainly to raise vaccines. Now you are aware that when COVID is present, we are basically looking at the presence of antigen in the sample which we are testing. So these kind of antigens are used for raising vaccines. If you look at the COVID antigen, it is a cell surface antigen, which is, we call it the spike protein, which has got two components, the S1 component and the S2 component. So antibodies, and they are able to induce immune responses. That's why antibodies are raised against these spike proteins. So the antigens are used for inducing immune response and to produce vaccines. Several antigens are used for a routine work in laboratories. For example, some of the antigens that are used for routine works are, one of them is bovine serum albumin. Bovine serum albumin, as you know, it is BSA. It is a 69 kilodalton protein. Another antigen which is commonly used is the hen's egg lysozyme. It has a molecular weight of about 15 kilodalton. Another antigen is tetanus toxoid. Tetanus toxoid, which has got a molecular weight of about 150 kilodalton. Then there is a flagellin, which is found in the flagella of a bacteria, that has a molecular weight of roughly about 40 kilodalton. Another example of an antigen which is used in laboratory is an enzyme glucose oxidase, is another antigen that has a molecular weight of roughly about 80 kilodalton. So these are some of the antigens which are commonly used in a laboratory for several experiments. So the purpose of this video is to give you an idea about different types of antigen which is naturally present.